Sir, we are going live now.
Yeah. Are my slides visible?
Yeah, it's fine.
Yeah.
A very good afternoon to everyone. Myself, Dr. Nagamani, on behalf of DDH 2020 training team and Director CSARDs Dr. G.N. Sastry, we welcome you all for the Vertical 1 session. Today, the Vertical first session will be delivered by Dr. Ramesh Sistla, Managing Director of Think Molecular Technologies. Dr. Ramesh got his PhD from IIT Madras in computational condensed matter physics, and he then changed his course and moved to the field of computational biophysics for his postdoctoral research at the Molecular Biophysics Unit in Bangalore. And he led the group with several impactful contributions in various drug discovery groups in different companies till June 2020. He recently founded Think Molecular Technologies, a molecular simulation company at Bengaluru, which aims to partner with biotech and pharma companies to bring greater efficiency to drug discovery using MD simulations. With this small introduction, I request Dr. Ramesh to deliver the talk on what can a fluorine do, learning from IRAK4 inhibitors. Sir, over to you.
Okay. Are my slides visible?
Yes, sir.
Okay.
Please go ahead.
Yeah. Okay. So good afternoon to everyone from India, and a good morning if anyone watching it from the western part of the world. Okay. So, and of course, hello to everyone appropriately elsewhere in the world, because this is an online event. We need to get used to the etiquettes of this. So, thanks, Nagamani, for introducing me. As he has already mentioned, this is my new baby, Think Molecular Technologies. So where I aim to actually, deal with only molecular simulations and further the drug discovery of biotech and pharma companies. Okay. So these are my contacts. Obviously, you can reach out to me since this won't be highly interactive, although at the end there will be some questions that the organizers will take for me. They will gather for me and I will go through those. But in any case, if you want to reach out further and discuss with anything, these are my contacts. So today I'm going to talk about what fluorine can do. Okay, and this is particularly in relation to the IRAK4 inhibitors. And all of the work which is presented in this, what I'm going to talk about today, is from this paper, the ACS Medchem letters that we recently published, where this has been revealed. Okay. So let me get on to the beginning of this. So let's talk about what happened here. There is just a fluorine. Okay, so there was this compound 20, which had a potency of 3.2 nanomolar on IRAK4. Ambit selectivity index, which roughly says what is the percent of the kinome that your compound is inhibiting, is about 7.8. Okay, so it means that it is inhibiting about 8% of the kinome. Okay, so if your kinome actually in this particular case we tested over and an ambit selectivity index of 8% roughly corresponds to hitting 32 kinases somewhat reasonably potently. So that's a kind of a no-no in a kinase drug discovery program. We want to hit as few kinases as possible. Although there are other examples of where the selectivity, despite poor selectivity, compounds have become drugs, but as we actually prosecute the drug discovery programs, we try to actually keep this ambient selectivity index as low as possible. And then how could we do that? So if you just see, there's a matched pair of this compound 20, which is compound 21, and these numberings are as per the as per what has been given in the paper. So it just has a solitary fluorine there, okay, and the incorporation of this fluorine actually cleaned the compound up on the kinome, such that we were hitting only 1% of the kinome, down from about 8%. Okay, so that's a remarkable selectivity that we get in this that we got in this particular case. Okay.
Sir?
Yeah.
Sir, we are. I mean, still, are you in the first slide? We are seeing your title slide only.
Oh, is it? Not just a fluorine? Okay, let me reshare it. Okay.
Yeah.
Yeah. Do you see it now?
Yeah. Now, I'm able to see the second slide.
Yes. Yeah, that's where I am.
Yes, fine. Yeah, now it is good. Yeah.
Okay.
Now it's fine, sir. Please go ahead. Yeah.
Okay. Yeah, just stop me if you are. I will mention this slide number just in case. Okay. And if it doesn't come, just tell me. Okay.
Yeah.
Thanks.
Sure, sir. Yeah.
Thanks. That's very helpful. Thank you. Yeah. So I hope you followed what I said. In this particular case, just a single fluorine incorporation at this position alpha to the amine actually gave us enormous amount of selectivity. And that's going to be the crux of our topic today, how this fluorine actually did what it is. But obviously, I want to introduce IRAC4 and also take you through how we arrived at 20 and 21 in the first case. So that is going to be the part of the story. Now, this compound 21, obviously, because after this I'm not going to talk about any biology at all. So I just want to state here that the compound 21 demonstrated dose-dependent efficacy in the psoriasis model of mouse. Like if you take the scaling or erythema, as you can see that at 1 mg per kg, it is showing about 27% efficacy down to about 64% efficacy at 10 mg per kg. Okay. Similarly for erythema as well. Okay. So this was a kind of a proof of concept compound for us. So now, with that short introduction to what this compound 21 is all about, let's get into what IRAC4 is and what is its role in inflammation. Okay. Now, TLRs, the toll-like receptors, are actually membrane-anchored proteins. Okay, and these guys, so you see these antennas on top, they actually recognize sequences, conserved sequences from different microbes. I'm on slide number 3, by the way. So, and they actually recognize the sequences from microbes. So therefore, whenever there is a microbial infection, okay, so the body actually recognizes some part of the microbial RNA sequence through these toll-like receptors. What they do then is, upon the binding of that sequence there, they recruit what is known as a mid-88 protein here. Okay, it is written as MYD88, mid-88 protein. Okay. And what the mid-88 protein does is it recruits IRAC4. Okay, IRAC4 has, other than the kinase domain, it has a domain called the death domain, which is an independent domain of IRAC4. So the death domain of mid-88 and that of IRAC4, they actually come and they form a huge scaffolding complex. Okay, which is known as the midosome complex. Okay. What happens within the midosome complex is, by bringing together a number of IRAC4 molecules, what it does is it is able to phosphorylate. IRAC4 gets phosphorylated, and upon phosphorylation, the IRAC4 then dissociates from the midosome, and then it goes on and recruits other IRACs, okay, like IRAC1 and things like that, which go on to phosphorylate the TRAF6. Okay, and then the TRAF6 then goes and binds to the TAC1, and they signal through the nuclear translocation factor I kappa B, and then the final result of that is actually production of inflammatory cytokines such as interferons or the interleukins. So thus, IRAK4 actually plays a central role in the production of cytokines, and thus is important in the innate immunity pathway. Therefore, IRAK4 has been a much worked upon target in the pharmaceutical industry for its role in autoimmune diseases. So, how do we read this particular pathway on in our drug discovery programs? So, particularly in this paper, in this talk, we will focus on the IRAK4 IC50, okay, which is a readout, an enzymatic readout that happens at this level. And then there is a cytokine interleukin 6 release, okay, the inhibition of which we measure in PBMC cells and the human whole blood. Okay, HWB stands for human whole blood. Okay, so that's briefly the pathway of IRAK4 and its involvement. And pretty much, although there are a number of TLRs, a number of TLRs actually converge their signaling on IRAK4. Therefore, IRAK4 is thought to be a very good target in immunology. So, with that introduction to the pathway, since this is a molecular modeling talk, let's get down in more detail to the what happens at the molecular level. Now, the act of phosphorylation, how it happens on the right hand side. This is from the PDB-ID 2PHK, which is a phosphorylase kinase actually. This is a very interesting structure in the sense that it's a ternary complex of the kinase, ATP. The red line you see here is actually the substrate, okay, with a serine there. Such that the interesting structure where the kinase has been caught in the act of phosphorylation. A somewhat old structure, but nevertheless, this is probably for the first time we actually saw the structural proof that a phosphate group was getting transferred over here. The aspartic acid that you see here, I hope you are able to see my pointer. I'm on slide number 4. The aspartic acid here actually chelates the magnesium because the ATP is actually Mg ATP. It is also chelated. It binds to 2 magnesium ions. So when the aspartic acid actually chelates with the magnesium ion, and that is what helps in the cleavage of the phosphoester bond, such that you have the alpha, beta, and the gamma phosphates, and the phosphoester bond between the beta and the gamma phosphates that is cleaved, and the gamma phosphate tumbles over to the substrate serine or threonine or tyrosine. Accordingly, it's known as a serine kinase or a serine kinase or a tyrosine kinase, right? Yeah, I'm not going into more details of that. But it is, so what happens when the phosphorylation, when a kinase gets phosphorylated, that actually results in the stabilization of this DFG group, okay? This DFG loop actually, which is present here, about which I will talk again in the next slide. So that loop actually is stabilized, and that confers an active conformation on the kinase, which facilitates catalysis. And most kinases, they are actually activated upon phosphorylation. Okay, we call a phosphorylation transphosphorylation when when a given kinase is phosphorylated by another kinase, what we call generally the upstream kinase. And when a kinase phosphorylates itself and becomes gets into an active conformation, it is known as an autophosphorylation. Okay, so this is the act of the phosphorylation. So what does IRAK4 do? How does it get phosphorylated? This is a very interesting paper that came in 2014, which showed that IRAK4 actually gets trans-autophosphorylated, by which we mean that if there is a single molecule of IRAK4 with ATP bound, it cannot phosphorylate itself. But one molecule of IRAK4 actually phosphorylates the other molecule of the IRAK4 and vice versa. Therefore, for this autophosphorylation to happen, you need two molecules of IRAK4. So therefore, it is also trans, it is also auto at the same time, because in a biochemical assay, you would actually see it as autophosphorylation. But structurally speaking, we are actually needing two molecules here. Therefore, this is what we call the IRAK4 gets trans autophosphorylated. Okay. So, and this is precisely what happens in the midosome complex, where a number of IRAK4 molecules are bound along with the mid-88. They are getting into an active conformation, and then the activated IRAK4 is then sent out to phosphorylate IRAK1 and then carry out other downstream processes. Right. So let's talk about what are the interesting and key structural features of the IRAK4 kinase. This is how it would look from a great distance. This is the N-terminal is on top in case of IRAK4. In general, that is like that. So this is the N-terminal and this is the C-terminal. As you can make out, the C-terminal is highly alpha helical in nature. Okay, you see the number of alpha helices. Whereas the N-terminal is actually a mixture of both beta sheets and there is a solitary alpha helix, which plays a very important role. If you zoom in a little bit, there is a region known as the hinge region, okay, which is actually very important for any kinase binding, okay. And this hinge is the one which actually, this is the hinge region, and that is what connects the N-terminal lobe to the C-terminal lobe. And this is where ATP makes a bidentate interaction. It makes two hydrogen bonds in this particular region, and that's actually a very important part of the kinase. Then, as I mentioned, in the C-terminal, there is a helix, solitary helix in the N-terminal region. What we call the helix C in general, what you see in green here. This helix C, this fellow actually plays a very important role in catalysis. Okay, suffice to say that I am not going into more structural detail of how it does that. If you are interested, do get in touch with me, or you can read other kinase papers. Right, there's a vast amount of literature out there. And then we have a glycine-rich loop. Okay, so which actually sits on the top of the ATP. This is made with a very flexible motif called GXG, where G stands for glycine. That gives extraordinary flexibility to this particular loop. Okay, so this is what interacts with the ribose of ATP. Then, as I mentioned, there is an activation loop. As I mentioned in the previous slides about the DFG loop. Okay, so the DFG loop is actually adjacent again to the ATP binding site. As I mentioned, the D on the DFG loop actually chelates with the phosphate. So the DFG loop actually also known as activation loop, starts with a conserved sequence DFG and ends with another sequence XPE. Okay, in between. The activation loop actually can span for a number of amino acids, about 20 to 30. The composition varies for different kinases, and this is what actually gives the substrate specificity. Okay, so when a substrate comes and binds to the kinase, for example, this particular sequence in IRAK4 will recognize IRAK1 and phosphorylate that, and also it does IRAK4. So therefore, that's the importance of the activation loop. Okay, and usually the phosphorylation amino acid also lies on the activation loop only. So those are the general features of kinases. Coming to IRAK4, it has a special residue, tyrosine 262, what we call the gatekeeper residue. Okay. It is known as a gatekeeper because it actually just precedes the hinge region, what I showed in the first slide, or the second, or in the second picture. So this tyrosine just precedes the hinge region. Therefore, most kinases have a back pocket, okay, which is adjacent to the ATP binding site on the right hand side, if you will, in the orientation that I showed you. So if you actually go to the right hand side of the ATP, so usually ATP has nothing to do with this pocket. But in case of IRAK4, because of the presence of a bulky amino acid like that tyrosine, the back pocket is pretty much abrogated in case of IRAK4. So this is a very interesting feature of IRAK4 inhibitors, IRAK4 kinase, okay, where we can take advantage of this particular amino acid, and you will see in due course how we actually exploited it. If you come to the front pocket of the kinase, you see this blue loop here. Okay, this blue loop is what is known as a Shellman loop. Not present in most kinases, but this is an insertion loop that is present in IRAK4 kinase, and that again gives a pretty unique character to the front pocket of IRAK4, because of which the importance of which you will see in the coming slides. So overall, there is a ligand binding site that is there in the interface of the N-terminal lobe and the C-terminal lobe. Okay, so this is a PDB that is published as part of this paper. You could see this. And as you can see here, this particular PDB, the ligand is actually bound in the interface of the N-terminal and the C-terminal lobe. So with that, let's get into the evolution of structure-activity relationship. And at every point, I will stop. I won't stop, but I will relate it to the protein structure. That's why I spent some time to give you an overview of what is the general characters, what is the general nature of the pocket, and what is the how does the pocket look like. So we will try to make rationalize the SAR that we see based on the pocket. So first, just to show you how the structure-activity relationship evolved. Obviously, we made a number of compounds, but we are only showing some of the glimpses of that. If you take 1, 2, 3, 4, the 4 compounds which are here, they're all in the same ballpark range of potency. For example, in 1 and 2, we had a big group, actually a phenylglycinol, what we call here, in the ribose pocket. Okay, this was making some opportunistic interactions with the polar amino acids. There, it was making a hydrogen bond. But we generally had very poor exposure with this particular group. So, which is when we pretty much got rid of this whole thing and replaced it with an isopropyl, as you can see in this 3. Okay, so 3 because of which what happened, because of the loss of the hydrogen bond that one of these hydroxyls was making, we lost the potency a little bit. Okay, but then what we saw in the IRAK4 was that there was a front pocket that was available with the Shellman loop that I talked about. Okay, so because we had a handle of amide here, we could actually do a lot of SAR in this particular region. So that was our, so after converting this phenylglycinol into an isopropyl, we actually focused on the amide library exploration. And in that process, it is actually our chemists who designed this compound, because there is a Shellman loop here, and there were some polar amino acids they were seeing. So the idea was to actually reach out to that pocket, okay, through a phenyl sulfone, phenethyl sulfonamide, what we call. And you see what this phenethyl sulfonamide has done is it picks up a hydrogen bond here, other than actually filling up this hydrophobic cavity in the front pocket. Okay. Just give me a minute. Yeah. So additionally, what you see here is that if you looked at 1, 2, or 3, you see again this is the same ambit selectivity index, which is the number of kinases with 33% activity remaining. Okay. Divided by number of kinases tested at a given concentration. In this case, These were tested at 1 micromolar. Okay, this is how ambit does. You could look up for ambit selectivity. Obviously, it is a little bit dated these days. Even you get IC50 measurements, a dose response for every one of those 400 kinases actually. Okay, so this is a little bit in somewhat about a decade back, or maybe half a decade back. It was people were generally depicting Si33. But these days, there are other ways of depicting a kinase selectivity. So 1, 2, and 3 pretty much hit about 10 to 11% of the kinome. Okay, out of 400, they were hitting about 40 kinases. But the incorporation of the phenethyl sulfonamide, which I said was a unique feature of IRAK4, so you see it pretty much cleaned up the whole kinome, and we were hitting only about 1% of the kinome. Okay, maybe about 4 kinases with this at 1 micromolar, right? So that was so that's not a general rule, but for this class of compounds, that seemed to actually work. Okay, but the phenethyl sulfonamide, this compound 4 itself, it suffered from poor metabolic stability, arguably because of the phenethyl sulfonamide. This phenethyl group usually is a bad actor in the metabolic stability, gets oxidized very easily. So therefore, our next focus was to actually, design for this front pocket and make compounds which replace the phenethyl sulfonamide. Okay. Yeah. I'm on slide number 7 now, which shows an SAR table of the replacement of the phenethyl sulfonamide. As you can see here, we actually got rid of the phenethyl sulfonamide, and we systematically went about replacing that. We did a very systematic assay starting with isopropyl, not isopropyl, ethyl propyl butyl, and then put a hydroxyl in the end. There's a primary alcohol and the tertiary alcohol, and so on and so forth. And in each case, the IRAK4 IC50 was more or less in good shape. Okay, so we were consistently getting good IRAK4 potency, but our whole blood potency was actually variable. Okay, and the liver microsome, the metabolic stability of these were also bad, arguably again because of the long alkyl chains. Okay, so that is where sensing the possibilities of oxidation that can happen on this tertiary alcohol, this fluorine was actually designed so that we have this fluorinated tertiary alcohol here, and you can see that the liver microsomal stability also improved from about 52% here to about 92% there. And we continue to get good fold selectivity against LCK, which did translate into pretty decent kinome selectivity. If not 1%, it was in the range of about 2% to 4%. Okay, so therefore this was pretty encouraging, from which we actually got this compound 12 that had pretty decent IRAK4 potency. It screened well in the profile, well in the PBMC and the human whole blood. When we actually it inhibited IL-6 release basically. It had decent metabolic stability, and the fold selectivity against LCK was also good. Okay, so that is where 12 was a very important compound for us. And before I go about what happened to 12, here is the rationale. We had a structure-based rationale. So in this program, throughout actually, molecular modeling team was working with the medicinal chemistry team. The amount of data that was being generated, we were rationalizing it at every point, and then making new hypotheses, which went back to the chemistry team. And some of those hypotheses were being made and tested. So this was a very, very gratifying effort for me, for the team actually. So if you look at LCK8, The left-hand side picture actually shows you the LCK, and the right-hand side picture shows IRAK4. Okay. Now, this is not a docking mode on LCK or anything. I have simply put the compound there, overlaid it there, just to give you a glimpse of how the LCK4, how the LCK, front pocket is like. As you can see, it is highly solvent exposed. There are polar patches, and there is a plenty of solvent here. You contrast that with IRAK4. This is the Sherman loop that you saw in one of the earlier slides as a blue loop. That actually almost touches another insertion loop called the alpha D loop, and that pretty much forms a very occluded front pocket, which is again a very unique characteristic of IRAK4. So because of this kind of a front pocket, this was our hypothesis, abiding hypothesis at the time, and it stood true. It stood true with time also. That whenever you had, the IRAK4 front pocket was more tolerant to hydrophobic substitutions like the gem dimethyl or the fluoro here, as opposed to LCK. And by the same token, by extension, a lot of kinases have a very exposed front pocket, shallow front pocket like this. Therefore, this front pocket was not tolerated in those, and that was our hypothesis of why this front pocket actually gave us, good selectivity against a number of kinases, right? So let's go back to compound 12. What happened to compound 12? So this is the general, this is the crystal structure of compound 12. You can find it in the PDB with 6VQL is the accession ID for that. I have enumerated the interactions. The pyridine nitrogen actually makes the hinge region interaction. Benzothioazole, which is a key part of this, okay, what we call the gatekeeper fragment, because as I mentioned, this is the hinge. To the right side of the hinge, when you see in this particular orientation, you have the tyrosine there. Okay, so the tyrosine is the gatekeeper amino acid, and your benzothioazole actually had a pretty productive, pi stacking interaction with tyrosine 262, the gatekeeper fragment, what we call. Okay, this is the gatekeeper, and this is the interacting one, what we call the gatekeeper fragment. So in the upcoming slides, I might be using this word gatekeeper fragment. Okay, if I use that word, that is what it means. Okay, and this is where you have the front pocket, where the fluorine, the tertiary alcohol, the fluorinated tertiary alcohol actually, obviously, since this is somewhat symmetrical, the X-ray, the electron density in the X-ray cannot pinpoint where the hydroxy is. But when you actually modulate, when you do a protein preparation, generally the hydroxyl actually points to a proline 266, which is part of the extended hinge. Hydrogen bonds which are present in buried pockets actually are a lot stronger, as we know, because of their dielectric considerations. So that's how this fluorinated alcohol also, gave a good boost to the potency. Okay, and this is the cyclopropyl group here, where which is highly tolerant. This is where we had the phenyl glycinol that I showed you in one of the earlier slides. So this guy actually interacted with the glycine-rich loop of IRAK4. Okay. So those are the interactions, and 12 was a very useful compound for us because we could show the proof of concept through that. And as you can see, it showed it actually inhibited the IL-6 release by about 96% at a dose of 25 mg per kg. So this data is from an LTA-induced acute murine inflammation model. So what was wrong with 12? Why did we not stop at 12? We didn't stop at 12 because 12 had poor exposure, and in general, you had to drive up the doses a lot to see consistent efficacy in long-term efficacy models. It had poor pharmacokinetic exposure. From our medchem intuition, as well as some biotransformation studies, what we thought was that this is the 2 position of the benzothiazole where the oxidation is happening, and which is actually resulting in a high clearance of this compound 12. So therefore, while the rest of the molecule was reasonably optimized for us, we actually trained our guns on this on a heterocycle campaign. Okay, in other words, to actually we gunned for a good gatekeeper fragment. This is a brief SAR. We did prosecute a number of heterocycles, of which we are just showing a very few of them here. If you see, so as we said, the 2 position of the benzothiazole was the culprit, so we actually methylated that. Okay, when the methylation happened, the liver microsomal stability was good, the fold selectivity was also good, but our PBMC, the IL-6 release was not inhibited in the cellular systems. Therefore, we went with and 17 and 18, not fragments that you would generally see in medchem explorations, but these were very useful compounds for us to actually show that this is actually where the exposure problem was coming from, because when we had these compounds, the exposure of the compounds dramatically improved. We are not showing the data here, but the exposure of the compounds actually dramatically improved. So therefore, this is where the soft spot was, and obviously these rings were opening, so we didn't actually prosecute these further. We moved to more enterprising, 6,5 fused heterocycles, like the pyrazolopyrimidines, pyrazolopyridines, and things like that. So what happened? When we had groups like this, as you can see, my green arrow says it improved, whereas red arrow says it deteriorated. So the traffic signal representation, if you will. So here you can see that as we went from this system to this system, definitely our whole blood actually improved dramatically. Okay, not dramatically, as actually from here to here it improved dramatically. Okay, but what happened? You see the SI33, which is the ambient selectivity index. Okay, it took a huge beating. Okay, so because of the front pocket, while we did manage decent selectivity on LCK, the new player that was emerging as an off target was actually c-KIT. Okay, so while 19 and 20 both had good whole blood potency and good potency in the PBMC assay, and their stability was also pretty decent. Okay, we took a huge beating in terms of the kinome selectivity. Okay, and I will come to how we actually address that. And pretty much each of these gatekeeper, was sitting in the same fashion, extracting a pretty good binding energy by its interaction with the tyrosine 262. Okay, because these were electron deficient heterocycles, so they actually had a good pi stacking with the cresol side chain of the tyrosine amino acid. So what happened? And now let us see. This is a visual representation. If you look at the supplementary information of this paper, we have given the ambient selectivity index, the ambient data on about 400 kinases for 3 compounds. So I have plotted this from that. There are a number of more elegant ways of plotting the kinome data, because here you are plotting the data on about 400 different kinases. So you want to actually you want the visualization to convey things to you in a more easier fashion. However, I have done it differently, and I will take you through this slowly. So on the x-axis, actually, we have the kinases, which means that Each point on this graph is actually represents one kinase. On the top here, if the point lies on the top here, it means there is no inhibition of that kinase. Okay. Whereas if the point lies, the more down it lies, it points to more potent inhibition of that particular kinase. And this is at a concentration of 1 micromolar. So now I have two lines here. One is the blue line and the orange line. So blue line is the compound 12. As you can see, it has fewer spikes. You can actually count them on your fingers. 1, 2, 3, 4, 5, 6, 7, 8, 9, maybe. Okay. So that was consistent with its. So if we are testing on about 400 kinases, it had a selectivity index of about say 2.3 to 3, something like that. Okay. Whereas my whole idea is to actually show you how dramatically compound 20 lost its selectivity on the kinome. That is by showing that there are a lot more spikes which are actually touching down for compound 20 as compared to compound 12. So therefore, compound 20 is a pretty bad compound. Okay, so which inhibits a lot of other kinases and which can potentially give rise to other toxicity associated with the inhibition of those kinases. Therefore, since we were actually looking at inflammation indication, our margin for safety is also much narrower here as opposed to oncology indications. That's also going down pretty rapidly. But therefore, this kind of a compound was a no-no for us. Okay, and our team actually, went about scanning the gatekeeper pocket further, because it was pretty clear that when you moved from benzothioazole to this kind of a pyrazolopyridine, or pyrazolopyrimidine, you had an erosion of selectivity. So definitely we knew that that is where the problem was. So structurally, what did it mean? Okay. And then, as I have already introduced in one of the previous SAR tables, c-KIT was actually one of the representative kinases. Okay, and what was common for c-KIT was that, along with a lot of others, was that c-KIT has a gatekeeper residue as threonine. I hope you remember. IRAK4 has a tyrosine in this place, which this our green compound actually it's a binding mode on IRAK4. So that guy actually stacks well with the tyrosine, whereas threonine actually has got a c-KIT has actually got a threonine in that place. So what happened when I modeled the compound 20 on c-KIT? Okay, what I saw was that this ring, okay, this had actually dramatically flipped. Okay. Such that this nitrogen elicited an interaction with this threonine 670 of c-KIT. Okay, and when we modeled, and this nitrogen additionally was getting another interaction, but this was again consistent with our observation that if I take you to 2 slides back, you please remember this particular nitrogen. Okay, what happens? You see the c-KIT fold selectivity. I'm showing you 19 where we don't have that nitrogen. So we have a lot more fold selectivity for c-KIT as opposed to 20 where we had that nitrogen. So that is actually losing a lot of selectivity on c-KIT, which means that it is this nitrogen that is the culprit. And what is that nitrogen doing? That nitrogen is actually making a hydrogen bond with this threonine. Okay, so as I mentioned already, while the green compound is actually 20 modeled on IRAK4, it has a very usual IRAK4 binding mode. I'm sure you are very familiar with that by now. Whereas on c-KIT, this was the revelation to us. This new interaction was actually a revelation to us. Okay, and this flipped mode of this guy actually, was what was causing it. Okay. And as I have already made this point, this hydrogen bond with threonine 671, that is what actually drove the c-KIT activity, as I just now mentioned through the data of the compounds 19 and 20. So another thing that actually validated the hypothesis was when we sent 20 for ambit panel. Okay. If you look at the, they actually test on various variants of the KIT kinase, the c-KIT kinase, okay, out of which one of the mutants is actually T671A, okay, where they mutate this threonine into an alanine. As you can see, and mind you, in this particular case, in the presence of the compound, how much c-KIT activity is remaining, we see, therefore, a lower number means it is actually, the compound is inhibiting c-KIT very strongly, and a higher number means it is not inhibiting at all. So when you looked at this particular mutant, what happened was that the compound fully lost selectivity, fully lost its activity. This compound 20 completely lost its activity against this threonine, the c-KIT mutant. That very strongly established that this is exactly what is happening in this particular case. Okay. So then what to do? Okay. Again, our very enterprising chemistry colleagues came up with this compound, the fluorinated version of this. Okay, and what happened when we had that fluorine? So that pretty much brings me to the title of this talk. Okay, one fluorine and then magic happens. You see, there is the whole table here. That's not very important, but you take a look at this guy, compounds 20 and 21. Okay, 20 you already know it has only 8-fold selectivity on c-KIT with a amide selectivity index of about 8. Okay, when you put a fluorine, it just cleans up. Okay, it was extremely selective against the whole kinome, one fluorine. Okay, so the incorporation of that fluorine was actually magical. Again, to show you a plot like that, you see here. The blue line actually indicates for compound 20, this guy, where this looked like a saint when we compared it with, not this guy. That was a different guy. Yeah. So compared to compound 20, you see compound 21, it is actually hitting one kinase here, and maybe another kinase there, but it is pretty clean against the whole kinome. Okay, so far so good. The data is good. It exactly gives us what we want. But why is this happening? Okay, so at every point, the molecular modeling team and the medchem team were collaborating very well. And very quickly, we actually came up with this hypothesis of what could be happening with fluorine. If you looked at, this is just to complete the matter, let's look at the binding mode of 21 on IRAK4. It is pretty identical to what we have seen until now, where this guy pyridine nitrogen makes the hinge interaction, the nicotinamide makes the extended hinge interaction, so does the tertiary alcohol here, and the fluorinated pyrazolopyrimidine also enjoyed a good interaction with the tyrosine 262. Additionally, in several of the structures, what we have actually seen a conserved water here. This is a water that's not only seen in our series. If you look at other IRAK4 structures in the literature also, this conserved water is there. So I think, although we are not shown it here, or we couldn't have any such hypothesis, generally making interactions with conserved waters, strong hydrogen bonding interactions with conserved waters actually can give you very good selectivity. Okay, that can happen. Okay, so maybe some of the selectivity we see in this case also stems from this fact. Okay, now if you looked at the c-KIT binding mode, obviously with the compound 20, it was in this particular, it was in this fashion. But with the compound 21, even the c-KIT binding mode actually flipped. I'll talk a little bit more about that. Okay, so if you looked at the binding mode of 21 and 20 on, so the 20 on IRAK4 is like this. 21, because of the fluorine, also assumes an IRAK4 like binding on c-KIT, but this is not a very productive binding because we have lost interaction with this threonine, and this compound is also somewhat more displaced. So therefore, this guy lost activity on c-KIT. That was our structure-based rationalization for this. Now, why did this fluorinated heterocycle flip? That is that's the part of the next couple of slides. I'll be done in another 2-3 slides. I think we are doing good with time. Yeah. So what we did is, so this was the IRAC4 binding mode of 20 and the secret binding mode of 20. Okay. So the blue one is the green binding mode is that of the secret, whereas the white binding mode is that of IRAC4. Okay. As you can see, the ring flip is very obvious. Okay. So, and what is in play? This dihedral is between this nicotinamide, this pyridine ring, and this gatekeeper fragment that is in play. So therefore, what I did is a dihedral scan around this. And what did I find? So this is actually a QM dihedral scan done with Jaguar. With a step size of about 5 degrees, and as you can see, there is one minima here, and 20 is 20 is actually the green. 20 is actually the orange guy, whereas 21 is actually the green one. Okay, so 21 is our fluorinated compound, so that is the green curve, whereas the orange curve is actually our compound 20, which is actually the desfluoro version of that. There is another local minima here. If you look for 20, this minima is only about a kilocalorie or 1.5, at the most 1.5 kilocalorie above the global minimum. Okay, therefore 20 could actually adopt this conformation more freely. Okay, so I hope you're getting this. Yeah. So if you looked at this, the dihedral here, that actually corresponds to a binding mode like this, an IRAC4 like binding mode, right? Whereas the dihedral here actually corresponds to a a flipped version, a flipped mode of this particular heterocycle. Okay, so therefore this dihedral, when it is about say 160 degrees or so, now for 20 could still do that because it had only about a 1 kilocalorie or 1.5, 1.2 or 1.3 kilocalorie above the global minimum. Whereas for 21, when you see that when you have a fluorine there, obviously that went up to about 4.5 kilocalorie per mole. Okay, therefore what happened is 21 could not take this sort of a a conformation any longer on c-kit, and it had to actually fall back to this kind of a binding mode. And in any case, 21 adopts this kind of a binding mode on IRAC4, but since this binding mode, this kind of a binding mode is unproductive on c-kit, we actually lost potency on c-kit, or in other words, we got selectivity. Yeah. So that's what I show in this particular figure, where when you had the fluorinated, the 21 on c-kit, it's not a productive binding mode, it is lying here. Therefore, by actually respecting the conformational preference of this compound 21, we actually came to a particular dihedral which was unfavorable for c-kit, and therefore gave us selectivity against not only c-kit, but against the whole kinase, all of the kinases which had serine as a gatekeeper, serine at the gatekeeper position. Because arguably we can say that on all of those kinases, this compound 20 actually adopts a binding mode like this, and that is why it actually inhibits a lot of those kinases, right? So to sum up, actually, this is what you see, the graphical abstract in the paper, okay, with the binding mode of 12 and 20 and 21. We actually, what we did is the structure-based design of IRAC4 inhibitors. We utilize the key distinguishing features of IRAC4 to build kinome selectivity to the compounds, such as the front pocket and the gatekeeper. We exploited these two pretty well with this series. So the first wave of the compounds, actually, they displayed selectivity against the kinome. What I showed in the first SAR table, they came through the front pocket interactions. But then they were with 12, which had a benzethiazole. This had a poor exposure, which gave rise to a heterocycle campaign. Okay. That's where we came up with a number of promising 6,5 fused heterocycles, which improved exposures and whole blood potency, but eroded selectivity. And then there was a single fluorine that addressed this whole thing. Okay, so what were our learnings from this whole thing? Small changes can be a big deal. As molecular modelers who work very closely with the medicinal chemistry teams, we should be the molecular match pairs. If you are given a huge database anytime let's say you actually inherit a program in which there is data for about, say, 1,000 compounds. The first thing any molecular modeler should do is look for matched pairs. Not only molecular modeler, any drug hunter for that matter, you should look for molecular matched pairs. Like this is a very good example of how one fluorine actually causes this activity cliff. Okay. The second learning for us was conformational preferences of these small molecules are enablers of interactions with proteins and they influence binding. Now, this is a very important statement. All drug designers should actually be very mindful of this. We should constantly ask ourselves, what is the conformational preference of my small molecule? Okay, in native state, not quite in the in complex with the protein, but in its native state, what is the preference of this molecule? Okay, because the closer the gap between a native conformation and the bioactive conformation, for a productive binding, you don't need to spend any energy to actually get into a productive binding conformation. Therefore, you don't lose potency there. Therefore, experimental crystallographic databases like the RCSB or the Cambridge database, small molecule database, are actually the best friends of drug hunters. Structure-based rationalization, even if done post facto, is very useful in further designs and impacts the programs. Because I'm sure as I was presenting and I was crediting our chemistry team with a lot of these, you would be thinking, were all these just afterthoughts, okay, what I showed through modeling? Well, not always so, because afterthoughts are important. Sometimes they were so, but it is equally important to explain a good piece of data that is generated by the experimentalists. So therefore, the more intensely that we work with them, it actually impacts the program in a positive way. And there is nothing like an afterthought, because what is your learning in a given thing, you can always use that in upcoming designs. And as we did in this particular case, obviously this is a very small part of our whole thing. And in future, when you read our publications, you would know how we use this data further. So therefore, even if it is post facto, you should do a structure-based rationalization, and that impacts the programs. Use every bit of data that is available to validate the hypothesis. As you can see, I picked up one small bit of data from the ambit selectivity of 400 different kinases. That is a single point inhibition. But nevertheless, that data actually gave me more confidence in the hypothesis. Therefore, we could, prosecute the hypothesis much better. I hope this rings a bell with you. When people say, "Oh, fluorine is just like a proton," you should show them this example. There are a number of examples out there where fluorine, although for its because of its small size, it is often said like, "Oh, it's same as a proton." But I think that's a lot of hand waving. Fluorine is not proton. Okay, so this is a very good example of that. So that actually brings me to the end of my presentation. I just want to thank these wonderful guys at NIST, led by Shastri, for conducting these training sessions. Obviously, this is not a main job to get out every day after day and go after different speakers and then, schedule this whole thing. Really, hats off to you. Very good job. Okay. And I want to thank my past colleagues at BBRC and BMS, and who are also and many of the co-authors on the paper. And yeah, still we're getting used to the tuning in part. So thanks for tuning in. Meanwhile, stay safe, be masked, and yeah, I can take a few questions.
Yeah, thank you, sir. Thank you for your wonderful presentation. So now I request Dr. Himakshi to ask a few questions regarding your presentation.
Sure.
Good afternoon, sir.
Hello.
So myself, Dr. Himakshi Sharma. So I'll be handling this question answer session.
Yeah.
So, sir, we have received few questions from students, few basic questions we received. So first question, sir, shall we start?
Yeah, we can.
Okay, sir. So first question is, IRAK4 kinase belongs to which class of kinase family?
So it's a serine threonine kinase.
Okay, sir.
Yeah.
So the second question is, what is the function of activation loop other than substrate specificity?
Yeah, so that is the most important function because if you look at kinases, there are 500 of them which are phosphorylating a number of other kinases as well as proteins other than kinases. So therefore, the activation loops most unique thing is actually causing the activation as the name indicates. It is responsible for the active conformation of the kinase. Therefore, such that the phosphorylation of the downstream substrates can happen. I don't know if this question is asked from a drug design perspective. If it is so, when the activation loop is unphosphorylated, the activation loop takes many different conformations, one of which we call the DFG out conformation. Okay, that gives rise to other binders like type 2 kinase inhibitors.
Thank you, sir, for the explanation. So third question we have, does all the protein kinase functions are exactly same?
Yes, I mean if you talk about phosphorylation as being its function, it is identical because no matter whether it's a serine kinase, serine threonine kinase, or a tyrosine kinase, or a lipid kinase, what it does is phosphorylation. It's only the a substrate that differs. Okay, in case of serine threonine kinase, the substrate will have a serine or a threonine, whereas the tyrosine kinase will have a substrate as a tyrosine, a substrate containing a tyrosine, which gets phosphorylated. Lipid kinases bind to a number of lipids and then phosphorylate them. So kinases are actually unique in the sense of they're phosphorylating enzymes, so they're phosphotransferases. So to that extent, yes, they all do the same thing, but they all do the same thing to different guys. So because of which a whole lot of diversity actually comes.
Thank you, sir. So one last question I will take. That is, the ligand which has been shown in your PPT are binding to ATP binding site or other pocket?
No, they're site. So as I mentioned, ATP binding site actually will have ATP will have a hinge interaction. It makes a bidentate hinge interaction with the bidentate hydrogen bonding interaction with the hinge. So therefore, even our compound has a strong interaction with the hinge. So therefore, our compound is actually an ATP competitive inhibitor. Yes, or in other words, we call them type 1 inhibitors also.
Thank you, sir. So thank you for answering the questions. So on behalf of DDH training team and DDH training team chairman, Dr. G. Narahari Sastry, we thank you for being with us and delivering a lecture. So sir, we will wind up the session here. I request all the participants, please be online at 4 p.m. for the vertical 2 session. Thank you, sir. Thank you once again.
Thank you very much for giving me this opportunity. Yeah, thanks. Bye-bye.
Thanks. Bye, sir.